I would like to show you and review a genetics of prostate cancer, and hopefully you see how it's important it is in diagnosis and in treatment. So just very briefly, obviously you all know prostate cancer is, maybe I should say that. It's a very common disease, and most importantly, it has a very different, highly variable natural history, and this represents a significant clinical problem. So, common disease, and 1 in 10 men with prostate cancer also die of the disease. So it's very, very important question how we can distinguish the aggressive cases from non-aggressive cases. What I also would like to highlight is that based on the latest studies, it is now proven that there's a very large contribution of heritable factors to prostate cancer. This is the highest of all cancers, so 58% is heritable. And so far, only a small fraction has been identified. Also, there's a very few risk factors known for prostate cancer. This is age, family history, and ethnicity. It's a complex disease, and our aim worldwide for doing research in prostate cancer genetics is to discover components of the genetic predisposition, which hopefully will aid, and is already aiding, risk prediction, targeted screening, and targeted therapy. So the outline of my talk, first I'm going to hopefully cover the most important things in the germline genetics, rare variations and common variations, then a little bit of review on somatic genetics, and then hopefully I can show you a few examples how we can translate our findings into the clinic for screening, prevention, and treatment. So just very briefly, this graph shows the full spectrum of genetic variants for any complex disease. And if you want to plot the allele frequency against effect size, common variants with large effect size don't really exist. The evolution sorted those out. Very rare variants with minimal effect size are negligible. However, our aim is to discover all the variations in this middle band. So rare variants with large effect size are usually targeted by linkage studies. Common variants with small effect size are being discovered by genome-wide association studies. I'll talk about this. And the latest advantages in technology now allows us to use next-generation high-throughput sequencing to find rare and also sort of moderately rare uncommon variants. So first about the rare prostate cancer genes. So our team and lots of other teams around the world use linkage studies. This is collecting big families, and we try to follow the inheritance of the prostate cancer cases within big families. However, it was fairly unsuccessful for prostate cancer and really lacked robust findings. So now we can say that no single susceptibility loci is slowly responsible for a large fraction of familial prostate cancer. So now we can accept that really prostate cancer is a genetically complex disease. It has polygenic inheritance. So what we found, it was an epidemiological observation that prostate cancer often co-aggregated in breast and ovarian families. And this gave us an idea to screen the 2 breast cancer genes, BRCA1 and BRCA2, considered as candidate prostate cancer predisposition genes. So in our first study, we screened a little bit less than 300 cases, and we established that about 2% of these prostate cancer cases, they were all young, had deleterious mutations in the BRCA2 genes. This was followed up by a much larger studies. Now we screened about 2,000 prostate cancer cases, and we adjusted this frequency to about 1.3%. It's again for young onset cases. The estimated relative risk now for prostate cancer in BRCA2 mutation carriers is about 8-fold. It's quite substantial. Similarly, we concluded included the contribution of the BRCA1 mutations to prostate cancer. It's about 0.5% the frequency, and the increased relative risk about 4%. But most importantly, the clinical implication of this, that men, particularly who carry a mutation in BRCA2 gene, have a reduced survival. And this is important. For example, in the UK now, mutation carriers with prostate cancer, BRCA2 mutation carriers, are offered PARP inhibitors in clinical trials. And I'll come back to this study, which was published recently by the Stand Up to Cancer American collaboration, where they also found a frequent alteration in BRCA2 genes, both in the germline and in the tumor. So another clinical implication, one of our clinical fellows concluded a study, which is really important again, in a radiotherapy cohort. Really, the BRCA2 mutation carriers had a much reduced survival. We also tried to connect germline genetics with somatic genetics. And in a relatively small study, we also showed that those men with prostate cancer having a mutation in BRCA2 genes also have a significantly higher number of copy number changes and structural variations. So this was the first study linking germline genetics to the tumor genetics. So after advancement in technology, now we are trying to find rare prostate cancer predisposition genes by sequencing. Our first study screened genes which were also in a similar pathway to BRCA1 and 2 DNA repair pathways. And screening our biggest and best families, we established that 7% of familial cases carried a mutation in one of these DNA repair genes. Again, the most frequently mutated was BRCA2, but we found mutations, draw attention to ATM, CHEK2, BRCA1, etc. The important implication again, that those men who carry a mutation in any of these DNA repair genes have a much more aggressive disease. This we defined by nodal involvement and or metastasis or death from prostate cancer. So currently we are analyzing the data from a much larger study. Now we extended the gene set to about 200 genes, and we screened 1,000 prostate cancer cases and compare the frequency of mutation in all these genes against controls. And again, what we showed that definitely the DNA repair genes have more frequent mutations in generally prostate cancer cases compared to controls. And this is at the moment unpublished data, but we found a set of genes which most significantly differentiate between prostate cancer cases and controls. The most frequently mutated genes are listed here: BRCA1, 2, BRCA2, ATM, and CHEK2. So, only 1 minute left. That's a bit alarming. So the big question again for us doing the genetic research to distinguish between aggressive cases and non-aggressive cases. Because the rare variants are really, we need really big numbers to conclude how important the rare variation in prostate cancer predisposition. We have to analyze gene sets or pathways. And again, just the newest data that not only DNA repair genes significantly more mutated, but we found a new pathway, angiogenesis. And this is again important because drugs are available for these pathways. So just quickly moving on to finding the common variations. This is covered by genome-wide association studies. And this is again not just for common cancers, but all sorts of disease. hundreds of variants have been identified. And for prostate cancer, we established a very large collaboration. This is called Practical. And then we're working with other common cancers as well, and designed a custom Illumina array to test over 500,000 SNPs. And this is the current map of all the collaborators in Practical, over 100 groups, mostly in Europe, also in America. And we're very much hoping to recruit the first group in South America here from Brazil. So very, very quickly, again, the latest analysis was a huge study including over 80,000 cases and compared the frequency of SNPs against controls. And we identified 65 new prostate cancer loci. Okay. So this study has now been submitted to Nature Genetics, so soon you hopefully can see it published. And this is just an illustration of the newly discovered variants. Fine mapping of these common variants is also important, and there's a huge effort. Basically, these variants we discover just tagging a genomic region. So the aim of the fine mapping is to do a more precise location for these variants and ideally identify the candidate genes behind these signals. And so these are two examples. I don't want to go into the details, but behind the original signal, we find many more secondary signals and candidate genes, which again, potential targets for therapy. So now, if we want to summarize where we are with the pattern of genetic predisposition to prostate cancer, we established a few rare variants with higher effect. There are a few which needs to validate it. Hundreds now, it stands at 167 common variants. However, there's no reason to believe that there has to be more in this middle section as well. And current studies are targeting to find all these moderate effect risk. So the summary of the prostate cancer predisposition, we identified about 40% of the heritability, 30-35% is covered by the common variants, and 5 to 6% by rare variants. And what is important that now, if we screen a man, particularly for the common variants, we can say that those who are at the top 1% of the risk distribution have a much elevated risk. But similarly, those who are luckily inherited the low-risk alleles have a much reduced risk. And again, it has a very important clinical implication. So if a man walks now in the clinic, and we have these studies ongoing in the UK and perhaps in the US as well, and has a family history, father died of prostate cancer at age 60, we can more or less chart now the risk for the prostate cancer at the certain age. Now, if on top of it we add the results from genetic screening, and this is all just predisposition, we can have a much more precise evaluation of the risk of these men. So I'm gonna just rush through the genetics of prostate tumor. Lately, our team and a lot of other big collaborations started to do big genetic studies of the prostate tumors. And they either sequencing the whole exomes, the coding regions of the genome, whole genomes, and often we take multiple samples from cases to follow the evolution of the tumor. And the aim is to discover, again, the potential drivers for the tumors, which then can be drug targets. TCGA, I don't want to go into the details. I thought I'd bring it to you here because you may have heard about it. It's a big American collaboration, not just for prostate cancer, but a lot of other cancers, and an integration of a lot of data. And they identified multiple subtypes of prostate cancer. Our group in the UK is part of the big International Cancer Genome Consortium. And just last year, we established a pan-prostate group. And so far, we have about 1,200 whole genomes worldwide, mostly from UK, Germany, Canada, and the US. And our aim is to understand the field effect of prostate cancer. We discovered that even in the normal prostate, there are mutational signatures which show that there will be a development of the tumor. We also follow the clonal evolution, and we published a few papers on the clonal evolution of the prostate tumor and also the clonal evolution of the metastasis. And very briefly, this looks a very complicated figure. This again, unpublished. This is the genetic mutational signature of metastatic versus primary cancer. And what we can show that the metastatic cancers have a much more complicated signature and much more frequent rearrangements, deletion, etc. Again, the most frequently mutated genes, and on the genetic background, you can see that those which have germline, those men who carry germline mutations, particularly in the DNA repair pathway, have many, many more mutations in their tumor. And it's important, the clinical implication, men with more complex genetics of the tumor have a much reduced survival. I'll just skip through this. And I wanted to bring the relevance to this meeting. Obviously, the genetic background, whether germline or the somatic, will drive personalized therapy. Particularly the DNA repair pathways are a very good candidate for therapeutic intervention. And indeed, in our institute, Prof. Johan de Bono's team showed that those men who have mutation in the DNA repair gene, whether somatic or germline, have a much, much better response to olaparib. And I just wanted to emphasize one more thing. Our institute also have a very strong drug discovery and cancer therapeutic unit, and we're working with them. So what we do in the genetics, we discover the landscape of the genetic landscape of the prostate tumors, and we overlay this with cancer, which is a cancer research and drug discovery knowledge base. And basically the idea is that we can find new druggable targets. In our team, we have a very strong translational cancer genetics program, and we have several clinical trials for targeted screening, and also we follow up healthy men who we recruit with genetic predisposition. And we have shown that indeed healthy men who enroll to our study develop prostate cancer much more frequently. They present earlier age and with more significant disease. And this is more or less the last slide, which I cannot go into, but the liquid biopsy, I'm sure you heard a lot about it, particularly in advanced and metastatic cancer, will drive therapy and hopefully guide personalized treatment strategies. So in summary, I hope I could show you, although very briefly I just rushed through a lot of things, that with the ever-advancing technology, genetic screening of men will identify those at higher risk, leading for early diagnosis. But also it's important to identify those men who are at lower risk, because we can save enormous amount in public health. Once we identify the early stages of tumor, hopefully we can apply precision medicine, and we can find the best treatment options based on the genetic background. And as you see, there's a lot of huge efforts and collaboration, and data integration is extremely important. We need big numbers to have conclusions, and eventually we hope to understand the evolution of prostate cancer in general, and hopefully this will lead to development of novel treatment strategies. And finally, I would like to acknowledge my team. I have an excellent team back at the Institute of Cancer Research, and the practical collaborators. It's essential to have these big collaborations, and I really hope that the Brazilian group will have a contribution in the future to our work. Thank you very much.